Today, I'll talk about the sequence database search and BLAST. First, I want to review what we have learned in previous lecture. What is the alignment basics, or why we are comparing biological sequences? Basically, two reasons. First, the paradigm in biology, similar sequence to structure further to function. And the second, from evolutional point of view, similar sequence often from common origins or common ancestors. For homolog detection, depending on whether they have common activities or not, homologs can be divided to orthologs or paralogs. And we also learned similarity and identity. And remember that similarity does not always imply homology. And also, non-homology is not always come from non-similarity. And for pairwise sequence alignment method, we learned the basic one, brute force alignment, the enumeration, and the dot matrix or dot plot. And the most important one is the dynamic programming, which is the good plus good is still the good, or the big problem can be divided into sub-problems. And in 1970s, there is a famous algorithm, Needleman Watch algorithm, developed for global alignment, increased the speed for pairwise sequence alignment. And in 1980s, with the development in biology, Smith-Waterman algorithm is developed for local alignment. And also in 1980s, the sequence of proteins and genes is already been increasing a lot. At that time, when people often to do is for the protein sequence I'm starting, is it similar to a known protein sequence? If so, can I use the function of the known protein to guide my experiments? So at the beginning, everyone was using the Smith-Waterman method, and everyone is happy with that. But with time going by and with increasing amount of sequences, the Smith-Waterman algorithm is already too slow. So fortunately, in 1990s, Stephen Austro and David Lippmann published the BLAST method, Basic Local Alignment Search 2, in Journal of Molecular Biology. And after that, in 1997, they again published gapped BLAST and PSI BLAST. Gapped BLAST allow insertions, PSI BLAST for multiple sequence alignment. So this BLAST method solved the problem of finding the most similar gene or protein in a sequence database. And these two papers, the one in 1990 and 1997, according to Google Scholar, has been cited. One is over 80,000 times, the other is over 70,000 times. This is among the top 10 in the entire scientific field, not only in bioinformatics. So because there are so many tools developed based on BLAST, and there are so many different versions, techniques, papers, and books published, but we only have limited time, so we cannot cover all aspects of BLAST today, but I will give you a brief introduction to its basic idea and the concept, and also will show a very simple example how to use BLAST at the end of this lecture. Okay, so far we have learned how to align two sequences using two dynamic programming algorithms, Needleman Watch algorithm and the Smith-Waterman algorithm. But rather than do the alignment pairwise, it's more often to search sequence database in a high throughput style. And the goal is to identify the similarity between the query sequence, whose structure and function are usually unknown, and sequence in database, whose structures and functions have been elucidated and annotated intensively. Local alignment can be used for database searching. The problem can be formatted as following. Given a query sequence Q and a sequence database D, find a list of sequence from D that is most similar to Q. The input is the query sequence, the database, and a scoring matrix based on which the alignment score can be calculated. And the output is the ranked list of hits, and the ranking is based on certain score. Next, let's estimate the time it takes to perform a sequence alignment using Smith-Waterman algorithm. If we have two sequences on the column and the row of a matrix, then the alignment score Sij can be considered as the index of the matrix elements. So the iteration of the formula can be converted into fill in the matrix cell by cell from left to right, from top to bottom. Then, for two sequences of length m and n, there will be a total of m times n entries in this matrix, or there will be a total of m times n grids. If we assume a query sequence has 150 amino acids, and based on the most recent release of UniPro protein database in 2019, it contains over 500,000 sequences with over 200 million amino acids, and on average, the sequence length in the database is about 359 amino acids. And assume our computer can run one operation in about 1 microsecond. Then we will need about 8 hours for one comparison. The size of amino acid database is increasing rapidly. The growth rate of nucleic acid database is even more striking. Here I show you the growth rate in the gene bank base pairs from 1982 to 2018. There is an increase in the order of 10 to the power of 6 in the base pairs. So for dynamic programming algorithm, to reduce the search time, the key is to minimize the number of grids that need to be filled in the dynamic programming matrix. In other words, try to calculate around the optimal alignment path, but not everywhere in the matrix, only around the diagonal direction. So BLAST is based on this idea. Specifically, BLAST use some heuristic method to first remove the database sequences without a strong local similarity to the query sequence in a quick initial scan. So the initial large database becomes a smaller database, and the speed increased dramatically.